I remember while I was a neurology resident reading an editorial by David Havler that was calling these genetic variants influencing disease severity as the holy grail of MS genetics. I couldn't really figure out if he was calling them the holy grail because he thought they were important or because he thought they didn't exist.
I remember seeing for the first time that Manhattan plot showing a signal that exceeded genome-wide significance threshold, and we were all ecstatic to see that clean result, unequivocal that we had found something. So those are the great moments in researcher's career when you say, yeah, it's all worth it. So it was fantastic.
Why do some people with the disease, as Sergio said, run marathons after 20 years and others are unfortunately in a wheelchair? Well, we now can say confidently that genetics is part of the answer. Hi, my name is Adil Harroud. I'm an assistant professor in the Department of Neurology and Neurosurgery at McGill University in Montreal, Canada.
Hello, my name is Sergio Baranzini, and I'm a professor of neurology at the University of California in San Francisco.
And I'm Brett Drummond. You're listening to the ECTRIMS podcast, the official podcast from the European Committee for Treatment and Research in Multiple Sclerosis. We've known for a long time that while MS isn't a classic genetic disease, it does have a genetic component to it. However, while many genetic variants have been identified over the past decades that relate to disease susceptibility, it wasn't until the middle of 2023 that research was published related to the discovery of a genetic marker that correlated to disease severity. My guests on today's podcast, Sergio and Adil, were authors on this groundbreaking study, which was published in Nature. Sergio, Adil, thank you for joining me.
Glad to be here.
Thank you, Brett.
So as I mentioned in the introduction, we've known that MS is a disease that has a genetic component to it. Prior to your study, can you explain what we knew about the genetic involvement of MS?
Sure. So MS, we've known for a long time that it has a genetic component, and this is from the 1970s where the first association with MS was discovered, and that was in the HLA region. So these genes that encode proteins involved in self and non-self recognition were primarily identified as being associated with MS, just analyzing a few families. And it wasn't until the mid-2000s where much larger genetic studies were performed, where we discovered the first non-HLA related genetic associations with MS. And 20 years later, now we know that there's about 230 associations with risk for multiple sclerosis.
Just to add to that, Sergio, we know of those 230, but we know that there are many, many more that we haven't been able to pinpoint yet. And I believe a big question over the past many decades was how do these common diseases, how do genetic contributions work for these common diseases? Is it a accumulation of familial forms with a single variant, single disease, single gene associations like we see for leukodystrophies? Or is it something different? And it so happens that it's very different, where we have thousands of common genetic variants that are not pathogenic in themselves, that are not sufficient to cause the disease, but that together explain up to, we say, 50% of the risk of MS, which is a very big share.
And so, I mean, you said there's many, many genetic variants that have been identified, more than 230 now that have been described in the literature, but all of these related to the risk of developing multiple sclerosis. And obviously, there's a really clear reason why we'd be interested in trying to understand those. But what made you shift away from that and be more interested in investigating genetic markers of severity?
So the genetic variants that influence susceptibility can tell us a lot about why people develop the disease. And people have used different ways of aggregating them to predict disease risk in individuals. They've also helped us understand biology, understand epidemiology. But what they can't tell us is who's going to do better or who's going to do worse. So people have looked at this question repeatedly, and in study after study, these risk variants were not found to be associated with things like disability or prognosis. And so, if our goal through the lens of human genetics is to help address the questions of disease heterogeneity and disease outcomes, and help explain them and understand the underlying biology through genetics, then we really need to shift our attention from studying risk to studying severity directly, because it seemed like these were different dimensions.
Absolutely. And so that was a big question in the field. Unfortunately, with the studies that the genetics of MS scientific community had done, we just didn't have enough power to identify those markers of disease progression. So it became really an opportunity to start to recruit patients and information to conduct specifically a study that addressed the issue of progression. So, which is what we determine an endophenotype. So, not looking cases versus healthy controls, but just looking within cases, what are the differences between people who develop the disease and then progress to, let's say, a wheelchair situation within 5 years versus those who might be still running marathons after 10 years. That genetic? That was the question that we set out to address in this study.
I remember while I was a neurology resident reading an editorial by David Hafler that was calling these genetic variants influencing disease severity as the holy grail of MS genetics. And I still think back to that editorial often, and I couldn't really figure out if he was calling them the holy grail because he thought they were important or because he thought they didn't exist. But I feel like at the time there was a little bit of both. And that was back in 2018, I believe, when the big IMSGC study on MS severity had just come out that you, Sergio, were involved in, of course. I remember asking people at that time that were giving lectures on MS genetics, well, what about severity? And their answer was, we don't know, but Sergio and Stephen just got a grant to study that. And that's how I got in touch with you to work on this.
It brings up a really interesting question that I was thinking as you're going through this. Considering we'd had so many years of looking for genetic markers and all of them were related to susceptibility, when you were launching into this study, and the comments, I think, from David there are pretty interesting in your interpretation of them, how confident were you that you might find something, or did you think that potentially these markers just didn't exist and that's why nothing had been found previously?
Oh, that's a great question. And actually, I think we went in with a healthy dose of skepticism. We didn't have any preconceived idea of whether we will find something or not. In fact, I think we were prepared to not find anything. But with the study that we designed, we were confident that at least if we didn't find anything, it would have been meaningful. So this is one of those things where a negative result would have been meaningful too. So when we found something, we were ecstatic because that will open the door for a lot of more science and potentially therapeutics. But we didn't know when we went in.
Yep. And so I think that it's sort of been touched upon a little bit in terms of why previous studies weren't able to find anything, and talking there about how this study was really designed to make sure that whatever the outcome, it was going to give a relatively definitive answer one way or the other. So can you talk to us just a little bit about how you went about conducting this study and how it was different from previous ones, and in particular the role of the two consortiums that were involved in this?
Yeah, so the two consortia are the International Multiple Sclerosis Genetics Consortium and the Multiple MS Consortium. So the IMSGC is a consortium that's been established for over 20 years and has really spearheaded all of the major genetic studies in MS. This consortium groups many centers from across North America, Europe, Australia, and outside. And they had collected about 15,000 people with MS with clinical and genetic data to be included in this study. And these were not just random cases. These were specifically enriched for people that were older and that had a longer disease duration. On average, they were 52 years old and had had the disease for over 20 years, with the notion that these individuals will have already declared their outcomes. We don't need to know if they're going to we don't need to guess if they're going to have a more severe or more benign disease course. We can directly observe it from the clinical follow-up. And then the multiple MS consortium is really a stroke of good fortune, isn't it, Sergio? If you want to explain a little bit how that fits in.
Well, absolutely. I think this is the kind of work that we feel cannot be done individually. So genetics is one of those research areas where individual laboratories are just unable to uncover or to unleash the kind of sample sizes that are needed to get statistical power to discover meaningful associations. And we learned this the hard way by trial and error, smaller studies which were coming back negative or inconclusive, I should say, because of lack of power. But we never would have known that that was lack of power if we didn't try and come together as an international consortium, pull together all the efforts, all the samples into a really statistically well-powered study. And then suddenly the associations started to emerge. So this is one of the great take-home messages, I think, and really gratifying from modern science, that modern science is collaborative, is group science, and only achieving these numbers is only possible by pooling efforts from many laboratories around the world. So we're really privileged and honored to be working with this fantastic group of people from all around the world.
Yep. And also a really important point of how critical it is that we have people living with MS engaged in the research that's going on and willing to contribute to these studies, because obviously without the large numbers of people being willing to contribute their data to this, these sorts of studies also aren't possible.
Absolutely. And the IMSGC was half of the story for multiple MS. This was an effort that was put together through European funding, led by the Karolinska and Ingrid Kokum's group. And they provided us with an additional 9,800 patients. So all in all, we had 22,000 patients available. And behind each of these patients, obviously, is a story and someone that's allowing us to conduct the research and share the data and so on.
Well, let's get into what the findings were, because the findings obviously were really exciting. So can you talk us through exactly what the key findings of your study were?
So I lost track who's next, but I'll take a first crack at it, and you correct me or expand. So the study literally consisted of gathering 10,000 DNA samples from people with MS, and we genotyped them. So we literally interrogated about 1 million DNA variants in each of them. And the first statistical analysis that was done is an analysis called heritability analysis. And what that does is it tells you whether the phenotype you're looking for, or you're looking at, in this case, disease severity, is in any way correlated with the genetic variation that you're seeing in general. And that analysis showed us that the heritability of the phenotype, in this case of disease severity, was in the ballpark of 13%, and that was really good news for a study like this, because it was in the same ballpark as the heritability of disease susceptibility, a little lower, but compared to other diseases, it was significantly higher, suggesting that we would have enough statistical power to detect variants if we looked for them. So that was the first thing, and I'll let Adil comment on the next step of that, which is the GWAS itself.
Yeah, I agree that heritability finding is key, because if you don't find genetic contributions, then there's no point in looking for genetic modifiers. And that 13% is really just what we can capture. And we know that there's a lot that we're not capturing. So, as Sergio mentioned, we've genotyped nearly 1 million variants, but then we were able to, through a process called imputation, expand that set to about 8 million common variants. So that's covering pretty much all of the common genetic variation across the genome, across all autosomal chromosomes. And that's what we can capture. So we know we're not capturing rare variants, and we know that they are likely to contribute to the phenotype as well. Like, they're likely to also impact MS severity. And so that heritability is going to be even more than 13%. And then the next step was trying to see if we can pinpoint specific locations within the genome that influence disease severity. And as a matter of fact, we did. So we identified one variant that was genome-wide significant. So what that means is that even after accounting for the huge burden of multiple testing across all 8 million variants, there was robust statistical evidence that this genetic variant associated with MS severity that we defined using the EDSS normalized for age, so the age-related MS severity score. And this variant, along with 11 other variants that showed a degree of significant association, but did not reach that genome-wide significance threshold that I just referenced to earlier, we took them forward for replication in the multiple MS cohort. So this means that our first analysis in those 12,500 led to some positive associations that we then brought forward into this second 9,800 patient strong cohort. And that genome-wide significant variant replicated independently in that sample. And one of the 11 also replicated independently. So we had quite robust statistical evidence that we had now on hand 2 genetic variants that were modifiers of disease severity.
So let me tell you a little story that happened during the process. So this whole analysis lasted over a year, and during that time, Adil, I, and Stephen Saucer, who is integral part of this task force during the whole process, we were meeting weekly during that period of time. And Adil at the time was in my lab in San Francisco, and then we will Zoom over with Stephen in Cambridge. And I remember that week after week, Adil was presenting with his quality control findings and imputation and additional quality control. And we will go from week to week with eliminating samples that didn't pass, or eliminating markers that didn't pass, and doing population stratification charts. And it was quality control after quality control. So it was really a year of almost just going through cleaning up and understanding the structure of the data. And then suddenly the day came that Adil said, well, next week we're going to have the results of the GWAS. And I remember seeing for the first time that Manhattan plot showing a signal that exceeded the genome-wide significance threshold, and we were all ecstatic to see that clean result, unequivocal that we had found something. So those are the great moments in a researcher's career when you say, yeah, it's all worth it. So it was fantastic.
Yeah, and I mean the data was really clear, and there were some really interesting things in that publication around the impact of that genetic variant and some of the clinical outcomes. And I think it was, from memory, dose dependent as well in terms of differences that you saw, if it was heterozygous or homozygous, with some clinical outcomes. Can you just talk us through those aspects as well? Because I think they really add to the story.
Yeah, that's an important question, Brad. To Sergio's point about there being moments that you look forward to and that you remember. I remember exactly what I was doing when I first saw that result, and it was trying to calm my crying baby. I remember that moment very well.
I'm sure they were tears of happiness based on your data.
I wish. So for that GWAS, we used, as I said, the age-related MS severity score. We had to do some data transformation to it to fit some, for the statistical model that we were using and so on. So it didn't really provide good interpretability. So to get that interpretability, we turned to outcomes that all people that work with in MS research will be familiar with, using longitudinal data that we had on a subset of 8,000 patients. So we had over 55,000 study visits spanning 14 years. And we looked at things like confirmed disability worsening, time to EDSS-6, rate of EDSS worsening. And it was quite striking that the association was consistent across all of these measures with a dose-dependent effect, as you just mentioned. Although it was also clear from those figures that the recessive group, so the individuals that inherit the risk variant from both of their parents, were at the highest risk. And for that confirmed disability worsening, we saw increased hazard ratios for time to EDSS-6. When we quantified the median time to EDSS-6, individuals that had both risk alleles had a 4-year shorter time to EDSS-6. And then we also saw a significant increase in the rate of EDSS worsening over time. And we saw that for both variants that we had identified in the GWAS. So that tells us that the approach we took with the age-related MS severity score was justified, because we're then able to replicate that across all of the outcomes that we're all familiar with and that we had access to.
So I just want to go back to one of the things that Adil just mentioned, because it's worth highlighting the fact that a single DNA variant could be responsible for achieving a milestone such as EDSS-6, which is the time point where usually a person with MS will need assistance to walk. And that if someone inherits that particular variant in a homozygote fashion, meaning both copies of it, then that person will achieve an EDSS of 6 almost 4 years earlier than someone who is heterozygote or someone who doesn't have that variant. So that is an extremely significant clinical effect for a single DNA variant. So we're that is on par of what one might expect from a platform therapy to be able to impact on. And we're talking about a single variant. We believe there are more to be discovered, and so therefore that's our excitement, that we think that we can address the issue of disease severity and potentially address the issue of progression with therapeutic options that can address these genetic variants that predispose to this worse outcome.
Yep. Well, I think that's why, the result in itself is obviously hugely exciting, being the first genetic marker related to disease severity. But I think it's also around what this may mean in the treatment and care of multiple sclerosis and of prognosis. And at MS Milan, and met both of you at the conference, and we discussed the fact that in almost every session I was sitting in, certainly people were bringing up this publication. It was across many different themes. So I would be interested in shifting to talk now about what these results mean for the future, for people living with MS, and for the research and treatment aspects of that. And in particular, things like, do you have a sense, even though understanding this is quite preliminary, but do we think this may enable more accurate prognosis at, say, a point of diagnosis? Is it something that will have the potential to impact on treatment decisions? Any comments around that?
Yeah, I think the prognosis question is really important. Just to put that in context, when we're talking about that one genetic variant that we're very, very confident in, and we haven't discussed this in detail, but we also have an abundance of pathology data showing that people that carry this genetic variant not only have more MS lesions, but also have things like reduced neuronal density and more evidence of axonal damage, and increased microglial activation. So we just have more and more data now confirming that this variant is important in the biology of the disease. But that is just, as Sergio said, one variant. And when we've mentioned before that heritability number of 13%, this variant alone explains less than 1%, which means that we're still missing a tremendous number of genetic influences that we just don't have the statistical power to, again, pick out from these 8 million genetic variants that are dispersed across the genome. So this is very much like searching for a needle in the haystack, except we can be very confident that there are hundreds, if not thousands, of these needles. So we found one, but we still have many laying in there. And it does tell us that there are genetic contributions. So why do some people with the disease, as Sergio said, run marathons after 20 years, and others are unfortunately in a wheelchair? Well, we now can say confidently that genetics is part of the answer. With respect to being able to prognosticate, right now, it would be really just for people that would carry this specific variant that we would have significant information to add. If we had the full catalog of genetic influences on MS severity, then we would be able to ask questions about prognostication. But for now, it's more limited. And what we need to get there is to increase our sample sizes and study more people, and potentially in parallel improve our phenotypes so that we have more precise measures of severity. And then we are able to start extracting more and more information from that genetic code. And so that's in regards to the prediction question.
And the other part is of course, very important, which is what might be the implications for therapeutics. And we now know that the development of disease-modifying therapeutics that have genetic backing, that have genetic evidence, are almost twice as likely to succeed. So the pharmaceutical industry is prioritizing heavily now, choosing their targets on whether or not there is genetic evidence. So we think that this opens up a whole new field for MS, which has been looking for alternatives for the progression. Now that genetic evidence exists, we think that this opens up the door for therapeutic development on drugs that might address the functions of the proteins encoded by the genes that are nearby the signal, and or their close genetic networks that are affected by it. And of course, more research is needed to fully understand the biology underlying this signal. But again, having the genetic evidence that progression and severity in particular is affected by these variants will, we think, steer a whole lot of research and development of therapeutic drugs.
And another aspect in relation to treatment is the fact that when we take the genetic associations with MS severity and those with MS susceptibility and compare them specifically with regards to where they fall in the genome in relation to genes. So if we think about the 20,000 genes that are encoded by the genome. Some of these genes will be more highly expressed in certain cell types and others in a different cell type. And so then we can relate our genetic findings to gene expression data that we can pull from a variety of tissues and cell types, and then start asking questions about how does the predicted expression of MS susceptibility resemble or differ from MS severity. And what we saw there is that when we look at MS susceptibility, the majority of the signal that we saw was in immune genes, so genes that are highly expressed in immune subsets. But when we did the exact same analysis with the same method with MS severity, what we saw was a CNS signature, so CNS tissues from the brain and from the spinal cord that showed the highest gene enrichment. And this tells us, first of all, that, again, they are different dimensions. It tells us also that it is what we interpret there as being CNS resilience that is the key determinant for MS outcomes. And it also tells us that we may need to get to this neurodegenerative dimension of the disease in order to meaningfully impact severity. And so that's another potential implication in relation to treatment, that neuroprotective drugs or perhaps remyelination, or not sure exactly what we're capturing there, but that's something that we need to be able to address as a community in order to really have a significant impact on severity.
An absolutely fascinating discussion, and we could talk for hours around this, but we should bring the episode to a close. Look, thank you both for all of your fantastic work on this and for joining us for the podcast today and sharing your insights.
Well, thank you so much for having us. And again, this is a group effort, and I really want to acknowledge all our colleagues from the IMSGC and Multiple MS Consortia from around the world. And a big thank you also for the patients who participated in this research. Thank you so much.
Thank you, Brett. It's been a pleasure, and always a pleasure to talk to you, Sergio.
Likewise, Adil. Anytime.
And thanks everyone for listening. Once again, I'm Brett Drummond, host of this podcast episode. I'm also co-founder of MS Translate, an independent resource centre that aims to simplify the complex wealth of information about MS for the greater MS community. The ECTRIMS podcast is produced and hosted in collaboration with two key partners, MS Translate and the MS Journal, a peer-reviewed international journal that focuses on MS, neuromyelitis optica, and other related autoimmune diseases of the central nervous system. The ECTRIMS podcast is supported by an educational grant from Novartis, Roche, and Alexion AstraZeneca Rare Disease. Educational grant providers have no input on the podcast series content. You can read more about the topic of this latest podcast episode and more on the ECTRIMS website, ectrims.eu. Do subscribe to the ECTRIMS podcast on your favourite podcast channels and follow ECTRIMS on X, formerly Twitter, LinkedIn, Facebook, and YouTube.